Yeah.
Howdy. So it's my pleasure to introduce today's seminar speaker, Dr. Lavanya Reddivari. Dr. Lavanya Reddivari completed her bachelor's in horticulture and did a master's in plant biotechnology in India. She received a PhD from Texas A&M University in this department. Currently, she's an assistant professor in the food science department at Purdue, focusing on plant food bioactive components and gut bacterial metabolism in health and low-grade inflammation-driven chronic diseases. With a goal to develop food-based strategies to encounter diseases such as ulcerative colitis and colon cancer, she has co-authored several peer-reviewed publications in the area of food and health and received several grants from industries, institutions, and USDA with a total funding of $3.2 million for her program. With that introduction, I'll let Dr. Lavanya continue.
Can you hear me okay? Thank you, Kishan, for the introduction. I'm excited to be here today in the department where I graduated from after a long time. And I really remember this classroom where I first TA'd Introduction to Horticulture, where my advisor, Dr. Miller, used to teach the class. And I do really miss him. And he was pretty instrumental in guiding me all through my career path. And still he's a role model, advising students especially. And I really appreciate the opportunity to share with you a little bit about our research program. Before I jump into the topic, I want to just give an overview of the long-term research goal of our lab. It is to develop food-based or plant-based dietary approaches for chronic colonic disease prevention. In that process, we want to understand how plant secondary metabolites in the fruits and vegetables, they influence the barrier function. When I say barrier, it is the human colonic barrier, which actually inhibits the translocation of the bacteria and toxicants into the systemic flow. And also we want to understand, when I say colon, it is the organ with relative abundance of several bacteria. And so we want to understand the interaction between these plant bioactive compounds and also gut bacteria and host immune system in exerting these health benefits. Recently, we are looking into polyphenol or the secondary metabolites and fiber interaction, especially improving the complexation, because naturally in fruits and vegetables, they are complex. The secondary metabolites are complex with the pectin and the cellulose. So we are trying to see whether we can increase the complexation so that we can improve the stability of these compounds and thereby there's health benefits. But today I'm going to focus on my talk and using the near isogenic lines, how we can use them to understand the single class of compounds, their health benefits in the whole food matrix. If you take a food matrix, a fruit or a vegetables, you have thousands of these compounds. And we want to understand a single class of compound in that food matrix. What's the health benefits of that? I'm going to tell you why we want to do that. So first, let's look at why chronic diseases. And if you really look at the prevalence is increasing, and in US alone, because of the chronic diseases, the economic burden is $2.5 trillion every year. Not only that, if you take top 10 leading causes of death, 7 out of those top 10 leading causes of death are due to chronic diseases. And I don't know whether you are aware of it, that July 10th, the 7/10 is a chronic disease awareness day to show that the 7 out of the 10 causes of death is due to these chronic diseases. Why plant-based diet? Okay, There are a lot of things we can do for a chronic disease prevention. Why a plant-based diet, especially the fruits and vegetables? Why? So there are a lot of epidemiological studies. I put a few here, that is recent ones, suggesting that there are multiple health benefits to the vegetarian diets or plant-based diet. And also these are very long-term, large studies. Now, what I mean by that is they have anywhere between 30,000 to 50,000 participants in these studies. And these are followed for 20 to 25 years. So these are a large studies suggesting that plant-based diet is inversely proportional to the chronic disease risk. And also, if you want to see what in these fruits and vegetables that is beneficial, and I'm going to take a reductionist approach here, kind of here, they're comparing plant-based diet and an animal-based diet. If you really look at it, macronutrients, micronutrients, vitamins may be similar. Of course, the type of the macronutrients differ, but they're all present in both. But we are going to focus on these non-essential phytonutrients that are only present in the plants. Okay, but they're not in the animal diet. Of course, so we are going to attribute these health benefits to those secondary metabolites. And again, there are some other studies showing that flavonoids, one of these secondary metabolites, are non-essential phytochemicals. Why I say non-essential? They're not essential for either the plant normal growth and development or human normal growth and development. But they are there in the plants. They can help abiotic and biotic stress tolerance. But in humans, they do have health benefits. So these are the two studies, and this is actually a recent study with 57,000 participants followed for 23 years. And during the 23-year period, they had 15,000 deaths. So they can really look at what are some of the risk factors for the mortality. And they do conclude that flavonoid intake is associated with lower mortality. So again, kind of pinpointing that these secondary metabolites have the health benefits. Of course, I have to agree with you guys, the benefits are not yet conclusive. And also these are association studies, like they're associated with reduced reduction. There's no cause and effect relationships. Yet with these studies, you cannot draw that direct cause and effect. But there are certain other cause and effect relationship studies and animal studies showing that these plant-based diets are health beneficial. And because of that, I think 20 years back when I started as a grad student, maybe 18 years back, there was a 5-a-day campaign that you need to have 5 servings of fruits and vegetables in your diet. But I think even decade of campaigning, spending $1 trillion, they could increase the serving size, maybe 0.8 servings. So the question is, how are we going to, I put that question for you. What do you think was the average intake of fruits and vegetables now in American diet? It's a number. I know it's low. I think Dr. Patil is excluded from it. Yeah, 2.2 to 2.8 servings. Its recommendation now is 5 to 8 servings, but it's very low. And I was talking to, asking grad students, the vitamin C content in the American diet. What is the source of, the top source of vitamin C content in American diet? I was surprised to see it was French fries. Yes, it is, potato has vitamin C content, but French fries, because when you heat them, it's a heat labile vitamin, but whatever is inside, that is the top source of in American diet. So I think we need to be increasing the servings of the fruits and vegetables, but maybe it's so many factors that influences that it's not going up. but how we can really increase the intake of these phytochemicals, that didn't work. What are some of the other ways? Of course, the other one which came up was the dietary supplements. As you kind of extract these compounds out, and you can see the shelves right now. You see every dietary supplement you can think about. And of course, they have their own disadvantages. Like we have thousands of compounds in the plant. Which dietary supplement you're going to take? Or sometimes they have a plant extracts. Are they really stable? So there are a lot of questions with the dietary supplement. And of course, the laboratory studies, and they do show that there is the benefits, but the concentrations they use are pretty high. And this is one I want to really stress. Almost always the benefits are found when the phytochemicals are consumed in the whole food, a fruit and vegetables, not as a pill form. And I can give the reasons why. When you extract these compounds in the native state, they're kind of glycosylated and acylated with other compounds. When you are extracting them out, they are not in their native form. And when they are not in their native form, their stability and the bioavailability changes. So you do not see maybe the same type of health benefits. They do have benefits, I'm not saying that one, but the concentrations you need to go are much higher compared to the whole food matrix. The other thing we wanted to do is that increase these phytochemicals through the breeding program. So you increase the content in the fruits and vegetables itself. With this one, again, as I said, there are thousands of compounds. Which are you going to increase in these fruits and vegetables? And there are some studies suggesting that, especially in potatoes, when they increased one class of compound, there is a reduction in the other classes of compounds. So you can understand there is a one source and you're shifting into one sink, the other sink is going to go down. And I think this is the study in VFSA, grapefruit, where they did the irradiation of the grapefruits, and irradiation increases the flavonoids, but the limonoids are going to go down. Again, there are so many ways you can increase these compounds, but you need to know what type of compound. Are you interested in the flavonoids or limonoids? Okay, which are going to be beneficial, or which you want to really pinpoint. So the question is, it's difficult to dissect in the whole food matrix that single class of compounds. So to address that, we are kind of use this approach near isogenic lines. Of course, I'm going to use maize as a model crop to show you. I know it's not an horticulture crop, but I'm going to show some data on the potatoes also, but it is kind of a tool I'm trying to present, because potato being a tetraploid, getting the near isogenic lines is not very, I don't say impossible, it's not very easy as that of the maize, which has been studied and its genome is pretty much sequenced. So we kind of opted for the maize. And there are some, I think, isogenic lines in the tomatoes with the lycopene content, where they have a gradation of the lycopene content. But we wanted to look at two classes of compounds, the flavonoids and also anthocyanins. And this is the pathway. I'm not going to go in detail with the pathway here. So these four genes are regulated by, in the flavonoids, they're regulated by P1, pericarp color 1 transcription factor. It's a mid transcription factor. And in the anthocyanins per se, these four are regulated by the 2 transcription factors. So we kind of created four isogenic lines. So A, B, C, D, I just put it in A, B, C, D there. And if you look at here, this is a mutant line where it does not produce any flavonoids or anthocyanins. And the B is the line which produces the flavonoids. And you can look at it, the accumulation of these ones are at the different parts of the column. So flavonoids accumulate in the pericarp, Whereas the anthocyanins, when we peel out the pericarp, you can see the colorless and they accumulate in the aileron layer. And this one, we are putting both of them together, the phlobophene and the anthocyanins. They're present in both pericarp, aileron, and you can see here, the pericarp, the phlobophene, they're reddish in color, and aileron, you have anthocyanins. So they're accumulating there. And we kind of characterized these lines with the spatial and temporal expression of all the genes that are responsible for it. If you want to look at it, my PhD student has a manuscript in Frontiers in Plant Sciences. So we characterized these near isogenic lines. And the macronutrient composition didn't change much among these lines. So I'm going to show you some data on when we use these lines to kind of look at their health benefits, especially we are using ulcerative colitis model. And I don't know how many of you know what ulcerative colitis is. One of the inflammatory bowel diseases, and where the chronic colonic inflammation is a major issue. And of course, you can see here, North America is a very high incidence of the ulcerative colitis. And it's, of course, it's not in the top 10 leading causes of death, but it is a risk factor for some of those leading causes of death, especially cardiovascular diseases, cancers, and respiratory diseases. And I want to give you a brief background on, because when I'm going to cover some of the biomarkers, I'm not going to understand. So I'm going to, if you can look at it here, it's a healthy gut and it's an ulcerative colitis gut. I want you to draw your attention on 3 points. First one is the bacterial dysbiosis. And if you look at here, you have a pretty good diversity of the bacteria. And when it comes here, you have a reduced diversity. And of course, there are some of the bacteria going up, which are considered to be associated with a disease condition, and some of the bacteria are going down, but there is a bacterial dysbiosis. And the second one is impaired barrier function. So this is the colon. This one is the mucus layer. And this is the second layer of defense, the colon epithelium itself. And if you really look at here, the bacteria, normally they're supposed not to get into the lumen. And in an ulcerative colitic condition, because this mucus layer is kind of compromised, and also if you see the epithelial layer, the barrier function is compromised. So the bacteria can enter inside and the toxicants can enter into the systemic flow. And the next, it is the inflammation. So it's going to trigger a lot of inflammatory markers, ILs and interleukins and NF-kappa-Bs. So the 3 things, there is a bacterial dysbiosis, an altered barrier function, and also the inflammation. So initially we wanted to do a quick experiment with phlobophene because there's not too much data on what the concentration of phlobophene, so this really works on inflammation or not. So we kind of used a CMC, which is a carboxymethyl cellulose, an emulsifier, which has been shown to induce a very low inflammation in the colon. So I do a lot of animal experiment with mice, and we are using like a 15% and 25% dietary intervention of these compounds. Here I'm using just a B line. A doesn't have any of these flavonoids, and the B has the phlobophene, because we know about anthocyanins. We did a lot of research and we know the concentrations and everything. So we are trying to look at the phlobophene. And if you look at here, as I said, the mucus is the barrier function, So when we feed the mice CMC, when we give it, the mucus is almost the green color, it's almost eroded. Okay. The control has a good mucus layer here. And when there is no flavonoids, you can still see there's a mucus layer here. You need to know that these do not have a flavonoids, but they have accumulation of phenolic acids. When we stop that pathway, the top compounds are still accumulating, not the bottom. So they still have phenolic acids. And here plus 15, we have much more, and 25. And you can see here mucus thickness. And when the mucus thickness is increasing, permeability is going to reduce because it's not permeating those bacteria and toxins into it. Of course, you need to have a selective permeability for the barrier, but not entering those toxins and bacteria. So next we used a little bit acute model. This is a DSS-induced colitis, and where we kind of used all the isogenic lines A, B, C, and D. And the first thing we normally measure the body weight. Normally when you have ulcerative colitis, there's a weight loss because they cannot absorb many things. So you can see with the DSS, the animals are losing weight. We have to sacrifice animals if they lose more than 20% of the body weight. So you can see this here. And the control, you don't see any weight loss. All of the lines, it doesn't matter which one. All of them are kind of pretty successful in reducing the, if you have the con there, either it has phenolic acids or flavofenes or anthocyanins, it doesn't matter. And colon length is the other one. The shortened colons is one of the hallmarks of the ulcerative colitis, because when you have the inflammation and the nix, it kind of shortens the colon. And again, of course, there is some differences, but still every one of them are pretty good at ameliorating this DSS-induced symptoms. And IL-6 is the inflammatory marker here. And of course, we do some significances coming here. Yes, the flavonoids, means phenolic acids and flavofenes, yes, they're effective, but seems anthocyanin seems to be much more effective compared to those two. And another inflammatory markers here, they don't do anything, and these are doing. So again, depending upon what you want to really look at, is it the inflammation or is the barrier function, depends upon which compounds you want to really and look at it. And I'm putting some more data on here. You can look at is the control A line. Of course, it's much worse here, the histology scores. And the anthocyanins and anthocyanins plus flavofenes seems to be much better in maintaining that integrity of the colon compared to the flavofenes and the phenolic acids. So again, are you looking at the histology? Are you looking at the inflammation? Which one you are want to, these compounds differ. But I want to just say that yes, we can pinpoint each and every compound, the effects, even in the food matrix with these isogenic lines. And let's look at a little bit on the bacteria, these compounds. Actually, in the ulcerative colitis, I said there is a dysbiosis of the bacteria. And maybe the dysbiosis can be reduced diversity or loss of beneficial symbionts or expansion of some of the pathogenic bacteria. So we looked at the bacterial, and this is the Picoye plot. And if you look at it, the DSS, it has the dysbiosis. But most of all other diets, they do reduce the dysbiosis. And kind of here looking at how different they are from the DSS versus how similar they are to the treatment or the healthy controls. I think most of the compounds are still helping in that bacterial dysbiosis. And again, looking at what type of bacteria that are upregulated or downregulated. Of course, these are at the phylum level. Again, you can look at it, the firmicutes to the bacteria, its ratio, it's considered one of the hallmarks of, like, whenever you have a disease status, this one goes down. So here, control has high, DSS goes down. And phenolic acids is not showing much effect, but all the other three seems to be ameliorating that situation. Again, the bacterial metabolites here, it's the same thing. The short chain fatty acids are high when these compounds are present. So now the question we wanted to address is bacteria, how the interaction, but the fundamental question we want to address is gut bacteria really essential for these compounds to act? Or if there is no bacteria at all, is these compounds are going to show any effect on this ulcerative colitis? So interaction, yes, I want to do other experiments, but just the fundamental question is bacterial metabolism is essential. Why? Because it has a lot of implications. If metabolism is very important, when there is a dysbiosis, at what stage of dysbiosis is these compounds are not going to act anymore? Or if the colitis is so severe that there are certain bacteria that are essential for the metabolism of these compounds, if they're not present, are they going to really act? So the question we want to ask is bacteria essential? So we put the mice on antibiotics for the whole study. So, and you kind of update this bacteria. And here I'm showing the antibiotics didn't have much effect on their body weights. They were pretty normal. And when we do the 16S sequencing, antibiotics, they reduced almost 98% of the bacteria is gone. So we know that kind of, we could have used the germ-free mice, and I do use the germ-free mice, but here we want them to have that immune system still. Germ-free mice, the immune system is a little different. So antibiotics, it's completely gone. And they do show, and when we put, this is the thing, when we use the DSS on antibiotic mouse, they're very, very, very susceptible to the colitis. We lost 60 mice in 2 days. They're all kind of like, they're so susceptible. So we have to adjust the DSS concentration so that they're surviving. They're very susceptible to the weight change in the antibiotic mice. You can see they're losing their weight much faster. And you can see their cecums are full of like internal bleeding because of the antibiotics. Normally it looks like this. Germ-free mice, it looks like this. Normally the cecums are enlarged. When the antibiotics with DSS, they're very susceptible. And I'm going to just show these compounds, they did not really act when there is no bacteria. You can see here, normally when there is bacteria, they're ameliorating those losses in the body weight. But here, they're not doing anything. And of course, I can show a lot of other data, it's the same because they're not acting. When there's no bacteria, these compounds doesn't show much of the health benefits. And maybe the reason we do show that with the metabolomics, we measure maybe around 100 of these compounds, gut bacterial metabolites. And you can see that whenever there is a presence of antibiotics, there's no bacteria. And you can see the concentration of these compounds, certain bacterial metabolites, they're very low. The metabolites of these anthocyanins and clobophene compared to where there's bacteria. So we are looking at whether supplementing these compounds is going to, again, I don't want to go to the isolated compounds, but supplementation of these metabolites is going to show those health benefits. Or looking at which bacteria are responsible for the metabolism of these compounds and supplementing them as a probiotics along with these compounds is going to show those health benefits. So that's what we are trying to look at it right now. And here I want to quickly give you maybe another few slides on the potato study. Here we want to look at, in the same class, we have anthocyanins. Different anthocyanins have any different effects. So these are not isogenic lines, of course, but the purple ones is the purple majesty has a malvidine and a petunidine, and red potatoes has pelargonidine. So they're different anthocyanins. And we are using the same thing. These are baked, okay, potatoes. We baked those potatoes and freeze dried them and put it into the mouse diet. And we are using the same mice model, the DSS-induced colitis model. And we are feeding them for 8 weeks, 5-week-old mice. And for 5 days, we put them on this DSS to develop the colitis and see whether they have any prevention of the colitis development. So first one is the colon length. As I said, shortened colons, DSS, yes, we do see shortened colons. Doesn't matter what type of anthocyanins, both the purples and the reds, they kind of, they're so similar to the control, though they have DSS, they kind of prevent that shortening of the colon. And the permeability, again, we look at the barrier function, the permeability of these we use normally one of the fluorescent dye, we gauze the mice, and it's supposed not to enter into the systemic flow because of the size of the particle. But if it is in the systemic flow, means your barrier function is impaired. And we collect the blood after 4 hours and look at in the blood if there is any fluorescence. So you can see normally there is a little bit of fluorescence, and when you put DSS, it increases. And again, both the potato diets kind of improving the barrier function. And also the MUC2 is that mucus secretion, the layer. We are pretty amazed with these anthocyanins. They actually, especially the purple potatoes, they increase that mucus secretion. And we have done few studies in everything, seems to induce the mucus secretion. So compared to DSS, where it goes way down, and potatoes, they improve that. Again, the inflammatory markers, I'm putting just one for each of the parameters. So, you know, DSS, the inflammation goes up. Purple potatoes, again, reduces this. Many a times the red, the pelargonidine, it doesn't seem to act on inflammation, but it is much better in the histological, the barrier function, and the MUC2, those kind of parameters. And again, we did the antibiotics to see whether anthocyanins in the potatoes also need bacteria. And you can see when we put them on the antibiotics, no bacteria, they do not act. So they are kind of purple potatoes, red potatoes, DSS, there is no improvement. Whereas when there's no antibiotics, that means the intact bacteria, you can see the benefit. I think this suggests that these compounds are metabolized by the bacteria in the colon, and then they are exerting those benefits in there. Or these compounds are utilized by the bacteria, and they are kind of improving the relative abundance of certain species of bacteria, which may be indirectly showing that health benefits. Maybe some of the reasons, but I think bacteria is kind of playing a role in these compounds, health benefits, at least in the chronic colonic inflammation models. Again, the intestinal permeability, you look at it, wherever the antibiotics are there, it's very high. Though there is a little bit, but there are no significant reduction in the things. The same thing with the inflammatory markers. But in the inflammatory markers in this study, we actually didn't see any increase in the inflammation when there is no bacteria. So when we put the antibiotic themselves reduces inflammation. So we couldn't really see that inflammation reduction with the compounds, because when there is antibiotics, even DSS, there is no increase in the inflammation. So antibiotics kind of like they're acting to suppress the inflammation. So we didn't see any effect here. Again, the MUC2 is that mucus layer, as I already talked about, goes down. When there is the antibiotics, we do not see much benefit with these compounds. Total. Again, we did a lot of metabolomics analysis to see how these compounds. I did not put what exactly they are, but you can see when there is no antibiotics, all these metabolites are present. But when we put them on the antibiotics, either they're not present or their concentrations are pretty low when they are on antibiotics. That means there's no bacteria and these compounds are not working on it. And we actually developed a single reaction monitor, MRM model, to kind of look at around 40 to 50 different bacterial metabolites, or the metabolites of these anthocyanins, all the secondary metabolites. And these are some of the ones in the potatoes that are significantly different between these two models. And this is the one of our articles got selected for a cover page on molecular nutrition and food research, and showing that the same thing as like a potatoes, when there is antibiotics, it doesn't show it still has that crypt loss and it doesn't show health benefits. Whereas when it has the intact microbiota, it's pretty beneficial. So this is the manuscript in the molecular nutrition food research and came end of last year, and it got selected for the cover page if you want to look at it. With that, I want to conclude that this anthocyanins, or maybe first I want to go with this, the near isogenic lines can be used as a model for understanding a single class of compounds and their health benefits in the whole food matrix, because our goal is improving the whole food, the fruits and vegetables with all those compounds, not per se the isolation of these compounds. And both the flovofenes and anthocyanins were shown to be effective against this experimental colitis, but in the intestinal barrier function and also modulating that microbiota. But the flovofenes seems to be a little bit less bioavailable compared to the anthocyanins. And we are trying to look at how we can improve their bioavailability by kind of complexing the nanoparticles with making them with the covalent bonding with those, so how we can improve the bioavailability of these compounds. So, but both the cases, either anthocyanins or the flovofenes, it seems to be the health benefits are dependent on the intact gut bacteria, and when there is no bacteria presence, they seem to be not exerting those effective health benefits. I put this one, Phenol Explorer. I don't know whether you have anybody seen it. It's a very good database. It's an European maintained, and it has all the fruits and vegetables and what's the bioactive compounds present and all the concentrations. And if you want to look at any processing effect, how it influences those bioactive compounds, it has the list. And of course, it's a growing database. And if you want to look at, okay, I want to eat this type of class of compounds, but the fruits and vegetables that are rich in it, and it has it. And also all the attached human studies and animal studies with those compounds. So it's a pretty good resource. I really like it if you want to look into, of course, USDA has it, but this one is pretty much growing, and you can select whichever compound or whichever food product you want to, and look at all the different classes of compounds and their benefits. With that, I would like to acknowledge all the collaborators and also the graduate students and the visiting scholars, and especially the funding agencies. I have almost, I had 3 USDA grants, and again, I will be getting 1 more, no, 2 more, one on co-PI and a PI. So continued support from USDA and a little bit on industry and other grants. And this is our group. Pinning is the one, the most of the work, and she's the PhD student. Most of the work I presented today, she's the one who did it, and she's my postdoc. Some of the potato work is done by her. I really like this one. So instead of a pharmacy, we want to have a pharmacy. Take 1 a day with tomato with a cucumber. So kind of if you have some problem maybe we will get here. And maybe we need to be eating a rainbow every day. Okay, with that, if you have any questions, I'll be happy to take.
You determine how much of the phenolics or the foods you give the mice to satisfy sort of a reasonable dose.
So the potatoes, when we put it in 20%, mice metabolic rate and human metabolic rate are very different. You cannot just extrapolate 20% to 20%. So if I want to convert it, it is maybe 1.5 large potatoes per day. So that kind of equates to 2 medium potatoes or 1.5 large potatoes. And the maize, same thing, similar. But we kind of normally try to rationalize based on how much of a carbohydrate consumption. If I put it in, it is a staple food. If I kind of like how much percentage of that carbohydrates can come from the potatoes or maize. So that's how. Yes.
Very basic question. Experiments what
These are baked and freeze dried. Yeah, they are baked potatoes. And we actually did a pig study with potatoes, chips, and baked. And I'm still kind of doing that one. It was like a 250-pound hogs. We had to like truckload of potatoes. We chipped them, and Van Runen Farms and Bakersfield, they grew the potatoes like so much. And we had 120 pigs, which are 250 pounds each. And we were feeding them purple potato chips and white potato chips and purple potato baked and white potato baked to look at how obesity parameters and inflammation and reversal and everything. It was a lot of work project. We are kind of analyzing and trying to publish. So pretty good results actually to looking at how these different. Everybody wanted the meat of those pigs. I don't know about that. The students like, oh, purple potatoes, we want the meat. So we'll see. Yes.
So the one question that I ask you said that in the colitis model you're using that the ones who keep with that condition have less diversity of flavor. What causes that? Is it the diet they eat or is it
one of the reasons they say is these people are very, very low tolerant to fibers and certain foods, which are shown to increase the bacterial diversity. And now they are not, and even the gastroenterologists recommend them don't eat those because they're kind of less tolerant to those. And that is the reason, the bacterial diversity.
So then how do you think, what is the mechanism by which this
So this and in those patients you are asking are normally?
Or no, because you showed some data that they are even in the mice.
So we did some in vitro studies putting directly these anthocyanins on different strains of bacteria, looking at whether they really help these bacteria to grow. And surprisingly, Bifidobacterium lactobacillus, some of these bacteria which are considered to be health beneficial, these compounds do increase the growth of these bacteria. And we looked at some of the E. coli strains which are considered to be pathogenic. Somehow they inhibit the growth of these bacteria, at least in the Petri dishes. We tried especially Enterobacteriaceae is one of the major issues in ulcerative colitis patients. The populations go way up, Enterobacteriaceae. And these compounds, at least in the Petri dish, they inhibit the Enterobacteriaceae bacteria growth. So yeah, that's the, I think we put it a drop and kind of grow the bacteria. Around the drop of anthocyanins, the bacteria doesn't grow, especially Enterobacteriaceae. But bifidobacteria, it grows on top of that wherever we are putting it. So we are trying to look at how it identifies it and why it happens. But I do not present, but they do inhibit. And we are thinking, I think maybe they have this enzyme system that they can use these compounds or metabolize these compounds. Maybe the Enterobacteriaceae it doesn't, but we do not know. Yes.
Good presentation. So how do you attribute that this effect is only because of extract?
Yeah, that's a question. So right now what we are trying is within the food matrix, of course, there are a lot of things. I mean, near isogenic lines for sure, but here with the potato, different anthocyanins, I do not know because the concentration of anthocyanins doesn't really explain my results. So what we are trying to do is take a potatoes and trying to extract these compounds out, and take that fiber. Many a times resistant starch is the one which I'm kind of a little bit because it has the health benefits. So I'm not really sure it is the resistant starch or the bioactive compounds. I cannot tease out that part of it. Where if I put a white potato, also it shows the effects because it has a lot of phenolic acids, much more than the mountain rose. So I'm not really kind of like, okay, it's antocyanin. That is the manuscript also, that's the question they ask, how you really say that it is the anthocyanins, not the resistant starch or anything. So we are trying to do one thing is kind of take the purple potatoes, take the anthocyanins out, use that all the resistant starch, everything as a control, and then the potato, purple potato, so that at least I'm eliminating whatever the, except for the anthocyanins, I have a control. Right now, I do have a potato starch in the control, but other than that, I'm not.
The question is, you really check the good bacteria and bad bacteria are also.
I think now the literature, you can't say whether it's a good bacteria, it's a bad bacteria, so you cannot. But at least the bacteria that are associated with the disease condition, yes, that is the Enterobacteriaceae. Whenever the populations go more than 4%, it's supposed to be always associated with the disease condition. So in this one, the Enterobacteriaceae levels are going down. Any other questions?
If you want to work with a implementary, it's not that favorable. So what is it?
What do you mean by they're not stable? Like a pH?
You want the concentration while you're doing.
I did a lot of in vitro cell culture experiments. A pH is the one. Of course, they are very sensitive to the pH changes. And if your media pH is going to be 6.57, and they kind of change into their which form they are present in. But the other form, it's not like it destroying the compounds, but you need to adjust your pH when you are doing the bacterial culture. But temperature and they're pretty okay. I think they're pretty because when we do mice pellets for the food, it does go through the heat treatment, and after we incorporate and we measure the anthocyanins, they are still there. But the pH you need to be really careful of.
Let us give a bit would like to.
Thank you.